Today, we would like to share information from Dr. Valentina Trinetta, who's Associate Professor in Animal Science Industry at K-State. Dr. Trinetta?
Thank you for this opportunity and welcome everybody. Today I will talk about the presence and characterization of Salmonella in finishing pigs and the journey of Salmonella from feed to fork. Salmonella is #1 of foodborne illnesses per year in the United States. Around 31% of outbreaks are every year linked to Salmonella, and swine pig are considered one of the main reservoirs for Salmonella. Traditionally speaking, we have been looking at the swine industry as different boxes where the farm was not connected to the slaughterhouse or the processing facility, and there was no connection between feed production and pig farm and or the final product that is going on the table of consumer. The Global Food System Initiative is one of the examples that highlight the need to consider any type of food industry related as a global system. And with this approach, we have been looking at the problem of why and how Salmonella is introduced in the pork supply chain from feed to fork. Specifically, we focus our research on Salmonella typhimurium and Salmonella monophasic, that is a variant of Salmonella typhimurium. And we were intrigued by these two serotypes because if you look at this table, these are the top 10 Salmonella serotype that are that were isolated and confirmed in human Salmonella infection. Around the same time, FDA, CVM did a survey where they went around for 7 years and they recovered Salmonella from animal feed. And you can see again in the top 10 serotype of Salmonella that were recovered from animal feed, we see again Salmonella typhimurium and Salmonella typhimurium monophasic. We did some previous work connected to this present project where we went around the feed mill in the United States and we tried to isolate and characterize Salmonella and in a feed mill environment. And what we found is that the same serotype that were among the top for human infection, we were able to find them also in a feed mill environment. So we came to the conclusion that even if the risk of salmonellosis is difficult to quantify from feed to fork, it cannot be excluded, and that feed mill represent a possible entry route into the human food value chain. Furthermore, the two serotypes that I highlighted previously are very dangerous for human because they are multi-drug resistant. And we know that multi-drug resistant bacteria can spread into the environment and arrive until our food. Therefore, we started this project sponsored by the Global Food System Initiative, where we wanted to understand what was the role of the pig. So the first objective was to evaluate the presence of Salmonella in finishing pigs. We were very fortunate. We selected 5 finishing farms across Kansas where we could go and talk with stakeholders and do our sampling. We identify 35 sampling sites per farm, both environmental and the fecal sample from the pig. And then we brought back the sample in our lab, and we follow a standard protocol from USDA FSIS for the isolation and identification of Salmonella. Very interesting. Here you can see the different, some of the sampling site that we swab. We found a lot of culture positive Salmonella in all the farm and in any sampling site that we analyze, there was Salmonella. When we continue our characterization and we did some molecular analysis, there was one particular farm, that was farm number 3, that had all the site confirmed for Salmonella. We did a little bit of history, and this, while all the other farms were never involved in any type of recall or outbreak, this farm was involved in a recall a couple of years ago. They did several interventions, but Salmonella is still there. That's kind of highlighting the problem with persistent foodborne pathogen. So anyway, this farm number 3 was full of Salmonella, and he gave us a couple of isolates. And in objective number 2, what we did was the characterization of these isolates by whole genome sequencing. We use an Illumina platform, and then we did some bioinformatic molecular analysis where we predict the serotype, we cluster the serotype in SNPs, and we evaluate the presence of virulence gene and or antimicrobial genes. What we observe, and this is the distribution of Salmonella serotype among farm, is that a lot of serotype like Agona, Steffenberg, the monophasic, the Typhimurium, that again are among the top 10 serotype for human salmonellosis, were also detected in farm and pig. When we cluster these isolates, they were matching with environmental, with feed, with pet food, with swine, and with other pork product isolate. So you can see here the phylogenetic tree where our sample are clustered with other environmental and other animal isolates. What was concerning and interesting for us to observe was that 52% of our isolate were clustered with clinical sample. And that means that we cannot exclude a porcine lineage. That means that also the farm and the pig have a key role in the transmission of Salmonella from feed to fork. The third objective of our study was the evaluation of how this pig can get Salmonella, and what was the vehicle where they got the majority of Salmonella. So we performed a study at the Large Animal Research Center at Kansas State University, where we did have 12 pig that we divided in 4 treatment. One was the control. These pigs were not treated anyway. And the other 3 groups where animals were inoculated with contaminated feed. So we did a gavage. With contaminated water, again, we did a gavage. And by air using a nebulizer. We contaminate them only, we challenge them only on day 1. And for the other remaining week, we did the daily health check. And we collect fecal, nasal, rectal, and environmental sample. Every day, these samples were enumerated. And at the end of the challenge study, we did necropsy, and we were able to do some histopathology in collaboration with the College of Vet. These are some of the results that we obtained from the detection of Salmonella on the 3 pig every time that we check fecal, rectal, nasal, and environmental swab. And you can see that the control didn't have any Salmonella positive, that the pig that were contaminated with aerosol, Salmonella aerosol, had mainly the nasal swab positive for Salmonella, and most of the other swab were negative, while this pig and the room where the animal were contaminated with Salmonella feed and Salmonella water, they did carry this Salmonella in the environment and in their body for almost all the challenge study. So here all the plus are when we detected Salmonella. At the end of the challenge study, we did necropsy, and our intent was to understand where Salmonella was going after it was ingested or breathed into the body of the pig. And we divided the tissue and the organ that we analyze in the respiratory system, in the immune system, in the digestive system, and muscular system. Not surprising, the animal that were infected with aerosol had all the lung that were positive for Salmonella, as well as some of the lymph node and the immune system. The pig that were infected with feed and water had almost all the time the tissue and the organ that we analyze positive for Salmonella. And very interesting, none of the muscle were positive for Salmonella. I say very interesting because when we look at outbreak, those are the parts that are usually found positive for Salmonella when people get sick. And so this is an evidence that probably contamination with Salmonella is happening through cross-contamination during the preparation of our meat pieces that then reach the table of consumers. We also, we also did some histopathology, and here you can see on the A, there is no lesion, and this was a muscle. Instead, when we look at the digestive system, we highlighted with this symbol all the lesion that we observe. So there was an increase neutrophils in the lamina propria. There is damage to the surface, to the epithelial surface, and the colonic glands contain a lot of debris. So what we concluded, that feces and area in contact with pig in a farm are more likely to be Salmonella positive, that feeder and water were not usually contaminated with Salmonella. And this is important to make the claim that nose-to-nose contact area are less likely to be the area where Salmonella is transmitted through between pig. That contamination of feed water and aerosolized particle can cause infection and colonization of pig, and that Salmonella was found in feces, rectal and nasal swab, tissue, and into environment. Therefore, colonization of pig is a big factor that can introduce Salmonella into the pork supply chain. Project outcome. We were able to support a master student that successfully defended in December 2021. We published 3 peer-reviewed papers, and we presented at 4 national meetings. We visited 5 swine farms in Kansas, and therefore we increased our interaction with stakeholders in the state. They know that they know now the job that we are doing, and the fact that we can help them if they have problem with food safety. And was very interesting because we established a collaboration with a professor in history of science. And this is a part that I didn't have time to explain further, but we were able to do a review on the history of Salmonella serotype in the swine industry. So that was very interesting. If we think ahead, what we plan to do is mainly to extend the research to other Salmonella serotype pathogen and animal species. We are curious to understand if we observe the same phenomena, for example, in broiler. We are gathering data and information to submit soon a proposal to the AFRI initiative. And I would like to thank the Global Food System Initiative, but also my research team.